This is Lecture 3, Functional Units of the Nervous System, and this corresponds to Chapter 3 of your book. Quick reminder that Homework 3 is due Sunday, this Sunday, the 27th at 11 p.m. And quick announcement, Homework 1, Question 6. A lot of people struggled with that question, and that's happened in the past too. The correct answer was that humans are apes. So Homo sapiens sapiens are apes. However, there was some confusing phrasing in the textbook, and there's some general debate in the field on using the terminology apes for humans. So because the answer is a little wishy-washy and may not be generally accepted by everyone in the field, I'm going to give everyone full credit for this question. One of your recommended readings this week is an article outlining some evidence that a certain diet may have speeded the process of evolution on the modern human brain. I want to note that this is an article from a pop science website, but this specific website and this specific author used good scientific sources, and the website is generally actually reliable. You do want to be really careful where you're getting your scientific information, especially from online news sites. It's usually better to read the source article, so the actual journal article, the published study if possible, but I understand that can be a little dry and take a lot longer. Just be careful. Even reputable newspapers such as the New York Times do tend to overinterpret scientific findings to create the best news story, whether intentional or not. It's usually not people who are trained in that scientific field who are reporting on the paper. So just be careful when you're consuming scientific news. Another recommended reading, this is an article about taking your brain back from social media. It's just coping skills to help you stay focused in studying and trying to complete work with the vast amount of social media temptations around you. This is based on evidence-based psychological techniques. So it is based on research, and you may or may not have access to the article. There might be a paywall, but the video that's attached has all the relevant information.
Okay, so section 3.1, which is cells of the nervous system. There was a debate in the early 1900s. Essentially, the two main actors were these guys, Golgi and Kajal. And Golgi believed that the nervous system was composed of a network of fibers, kind of like a nerve net where information flowed through like water. And Kajal came up with the neuron hypothesis, which is what we now know to be true. He believed that the nervous system was made of discrete cells called neurons. And Kajal also kind of amazingly figured out that neurons began as simple structures and became more complex as an organism became more mature. So we now know that's true, and that the human nervous system contains approximately 86 billion neurons, and there are different types of neurons that have specialized functions. The basic structure of the neuron has 3 divisions. The dendrites, which are these branching extensions like a tree branch up at the top, and this part of the cell gathers information from other neurons. The cell body, also called the soma, is the core region. It contains the nucleus and some other organelles, and it integrates the information received from the dendrites. And then the axon, neurons never have more than one axon, but they might have many dendrites, and the role of the axon is to carry information onto other cells. So this is a little bit more detailed picture. You can see that the up at the top, the dendrites, on that C picture, the dendrite is connecting with the axon from another neuron. Then the information comes down to the soma, where the nucleus is located. Nucleus kind of condenses the information and sends out information to other cells through the axon. If you look down here at the bottom, you can see that the axon terminates in kind of, it does branch out a little bit and terminates into an end foot, also called a terminal button. And then the terminal button synapses with the dendrites from other neurons. Okay, so looking at the basic neuron with a little more detail, up at the top we have the dendritic spines, which are protrusions from the dendrite that typically is the point of contact with other axons. And a neuron can have thousands of dendritic spines. The more spines, the more communication it has with other neurons. The axon hillock is this area right below the soma where the axon connects to the cell body. And this is where the action potential begins, which we'll talk about, I believe, in the next chapter. And then there's the axon collateral, which is like a separate, another branch of the axon. Teleodendria are the end branches of an axon, where which terminate in the terminal button, which, as we learned before, is the knob at the tip of the axon that conveys information to other neurons. And importantly, the synapse is the site of information transfer between neurons. This is usually between the terminal button of one neuron and the dendritic spine of another neuron. And that's something we're going to talk about in more detail in the next chapter as well. Just a brief, this isn't required information, but Golgi, the guy from the debate in the 1900s, came up with a staining technique that you might see when you look up pictures of neurons. It's this kind of beautiful amber color. And then more recently, I think some scientists have come up with this rainbow cell visualization technique, which can stain or color individual neurons that have different functions as different colors. So all the neurons with a specific function or type might be colored yellow, all the neurons of another type are colored green, and it makes these really beautiful pictures that are also functional and that they show us how neurons are related to each other. And here's some more visualizations showing possibly some white matter tracks up here on the top right, and then bunch of somas, bunch of neuron bodies over here. Okay, so neurons are the information processing units of the nervous system. Most behaviors are produced by groups of hundreds or thousands of neurons. So individual neurons don't typically have their own individual function. There used to be this idea that there's like a grandma neuron. So in some of the memory areas of the brain, people thought that there was a single neuron that encoded the memory of your grandmother. And I think in this particular research group that proposed this idea also thought there was a Jennifer Aniston neuron, because this was back in the 90s and when she was a little more relevant. So they did this study, and they believed that people who recognized Jennifer Aniston's face were encoding that in a single neuron. We now know that that's very likely not true, and overwhelming evidence indicates that groups of cells and networks of cells actually encode any even single piece of information. And so neural networks are really what's doing the jobs in the brain. So functional groups of neurons which are connected over wide areas of the brain and the spinal cord. And the connectome is a map of which areas of the nervous system have connections to other areas. In humans, this is not complete. We're not sure which areas, we don't have a complete map of which areas project to other areas, but it is an ongoing project. If you're interested, there's a link to the Human Connectome Project. And one of the types of visualizations, or the main type of visualization that is used in mapping the connectome, is these diffuser tensor imaging, or these images, yeah, I guess it's DTI images, which form these just really beautiful kind of rainbow colored visualizations of white matter tracks that connect to other areas of the brain. Neurons change their shape continuously. They grow and they shrink, and you can watch this YouTube video of an actual neuron that is changing, that is connecting to other neurons. In the picture here, you can see that a newborn might have a certain number of neurons, and then as an individual grows older, the connections between these neurons increases. There are new neurons created throughout life, and some behavior dependent on new neurons. However, within the central nervous system, the brain and the spine, most of those neurons are there when we're born and are never replaced. So this is one of the reasons why damage to the central nervous system is so serious, that because we're not able to create new neurons readily. Okay, there are three basic types of neurons that we're going to talk about. The first is the sensory neuron, which carries information from the body, or from like on the body, like the skin, or within the body, to the central nervous system. And if you remember the term afferent that we learned, I think in the last chapter, sensory neurons are afferent, meaning that they bring information from the periphery, from the body, to the central nervous system. There are two main types of sensory neurons. The bipolar neuron, which is found in the retina of the eye and sends information to the brain. The bipolar neuron is a pretty simple looking neuron. It only has one dendrite and of course has one axon. And these little green guys right here are bipolar neurons in the brain, in the eye, sorry. And then somatosensory neurons are the other type of sensory neuron. They project directly from the skin or the muscle to the spinal cord. Very long projections are possible. There's I guess a nerve, so a group of somatosensory neurons that run from the tip of the big toe to the base of the brain, which can be around 2 meters in humans. Most of that length is the axon, so it's very, very long axons in those particular somatosensory neurons. And this is a picture from a microscope of somatosensory neurons. Interneurons are the second type of neuron. They are only located in the central nervous system, so the brain and the spinal cord, and they relay information between sensory and motor neurons. So you can see maybe you can see why they're called interneurons. They are inter or in between neurons, kind of relaying information between. They're also called association neurons. Within interneurons, there are several types. One is stellate neuron, which means stellate means like star. So they are located in the thalamus, which, if you remember from last chapter, is like the switchboard of the brain. They are star-like in appearance and have many dendrites. And this picture down here on the bottom left is stellate neuron. Pyramidal neurons carry information from the cortex to the rest of the central nervous system, and there are two distinct sets of dendrites within pyramidal neurons. They're called pyramidal neurons because they're kind of pyramid shaped, so this second picture right here. And then Purkinje cells are located in the cerebellum, and they carry information from the cerebellum to the rest of the central nervous system. And they have a huge amount of dendrites, very, very dense dendrites. They look like little bushes. This picture of the map over here I thought was just a nice metaphor that somebody came up with, where they were talking about interneurons kind of being like the access road or the on-ramp, off-ramp between motor neurons and other motor neurons or sensory neurons. So they're kind of these in-between cells that create connections between motor neurons or sensory neurons. And finally, we have motor neurons, which send signals from the brain and spinal cord to the muscles. So they're efferent neurons, meaning that they start in the central nervous system and send information to the body. These typically have long axons, large soma, or cell bodies. Some of the longest axons of motor neurons in the human body reach from the base of the spine to the big toe, and these can be over 1 meter long. So you can see in this picture over here, this is a motor neuron, and it has quite a big cell body. So the three features of neural networks are that they have input, there's association, so interneurons are working, and they have output. Neurons with big cell bodies have longer projections in general, so they're engaged in processing information over large areas of the body, and neurons with smaller bodies have shorter projections in general, so they're more engaged in local processing, so maybe just processing within one area of the brain, for example. Excitation and inhibition is kind of the language of the brain. Neurons communicate through binary signals of yes, no. You can think of it as like ones and zeros, but we call it excitation and inhibition. A cell sums up the excitation and inhibition signals that it gets from its dendrites and sends the message along to other cells through its axon if the excitatory signals are greater than the inhibitory signals. So it's kind of this democratic process where it just tallies the votes and then decides if it should send the signal along. Okay, so glial cells are a different type of cell. They're not neurons. And they support cells in the nervous system. So they're kind of like the helpers for neurons. They do a range of functions like binding neurons together, providing nutrients, protection, helping when there's injury, helping with recovery. And very importantly, they do not transmit information. So neurons transmit information. Glial cells support this process, but do not themselves transmit information. There's about an equal number of neurons and glial cells within the nervous system. Scientists are increasingly recognizing the importance of glial cells in the nervous system. Before, people considered them to just kind of be filler, and more people are realizing that glial cells are extremely important in a lot of the in figuring out what's going wrong in diseases and etc. One example of this is that so Einstein's brain was preserved, and there's been quite a bit of conversation about what is different about his brain that might have allowed for him to have this such a higher level of intellect than the average person. And one hypothesis has been the fact that there were increased numbers of glial cells overall in Einstein's brain, but particularly in this area of the parietal lobe, known as the association cortex. This can't be proven, obviously, because we have a sample size of 1. His brain was compared against, I think, 11 other people of a comparable age to him. So there's some indication, but it's not can't be proven. And the 5 major types of glial cells are what we're going to discuss in the next few slides. The first type of glial cell, ependymal cells, produce and secrete cerebrospinal fluid, CSF, which fill the ventricles, which, if you remember, are those holes in the brain. CSF acts as as shock absorption. If the brain gets hit, it carries away waste products, maintains the temperature of the brain, and also provides nutrients to the brain. Another type of glial cells is the astrocyte, which is a star-shaped glia, and it provides structural support within the central nervous system. So it kind of holds neurons in place, holds neurons away from blood vessels, and also keeps the brain lining in place. It provides a pathway for nutrients to reach neurons. Astrocytes also secrete chemicals to heal injured neurons, and they contribute to the blood-brain barrier, which is a very important protective partition between blood vessels and the rest of the brain. The reason that blood vessels need to be separated from the rest of the brain is that this prevents toxic substances that might get in the blood system from damaging the brain. So the brain is very sensitive. Other areas of the body might be able to recover more quickly if they're exposed to toxic substances, but the brain, of course, is not as able to recover. The downside of the blood-brain barrier is that it can be really challenging to get certain medication, or to get medications to the brain at all. This is particularly a problem when there's like swelling or infection in the brain, and antibiotics can't get into the brain. So an infection of the brain actually can be extremely serious just because of the difficulty of getting medications to the brain. And that's all due to the blood-brain barrier. Okay, microglia is the next type of glial cells. They originate in the blood as part of the immune system and actually migrate up to the brain through the blood. They engulf foreign tissue, eat up dead brain cells, and they provide growth factors to damaged parts of the brain. oligodendroglia and Schwann cells are both types of glia that create the myelin sheath. And the myelin sheath is a very important part of the cell that it's like a fatty covering that insulates neuronal axons, and this actually increases the speed of information transfer. So insulation on an electric wire increases the speed of electricity going through the wire, and in the exact same way, the myelin sheath increases the speed of information passing through the axon. So you can imagine when we take this up a level, faster information going through our axons means we process information more quickly. Multiple sclerosis, or MS, is a disease you've probably heard of, and this one is caused by demyelination, so the atrophy or dying away of these myelin sheaths in the brain cell or spinal cord, and this leads to a number of issues in the motor system, behaviorally, emotional deregulation, lots of problems. Oligodendroglia are in the central nervous system, while Schwann cells are in the peripheral nervous system. So they both essentially do the same task, but do it in different nervous systems. And you can see up here the way that the Schwann cell forms the myelin sheath is it kind of like wraps itself around the axon literally, and then creates this outgrowth of the fat or the lipids to surround the axon, and then it keeps this up, tends it to make sure that it's healthy. Okay, and this is just an overview of those 5 types of cells that we discussed. Okay, so repair in the nervous system. In the peripheral nervous system, microglia and Schwann cells help to repair neurons, and so there's a figure of this to the right. In the central nervous system, repair doesn't really take place very effectively, and regrowth may be inhibited. So damage to the central nervous system, as I mentioned before, can be very serious. Okay, so moving on to section 3.2, which is the internal structure of the cell. I'm going to cover this very briefly and in minimal detail. This is important information, but there isn't time to cover all this information in depth. I think it's just a little too much for a 200 level class. So I'm gonna, in my slides, I'll give you the information that I think is most important. Please don't stress about knowing much more than what's on the slide. Okay, so first, the ions that are most critical to neuronal communication, the ones that might come up in later chapters, are sodium, potassium, calcium, and chloride. You don't need to know much more than that right now. The cell membrane is rather important, and we'll talk about it a bit in the next chapter as well. It's the double-layered outer wall of the cell, and it regulates what enters and leaves the cell. So proteins, there are proteins that are embedded in the cell membrane, and they act as kind of like gatekeepers to decide what goes in and out of the cell. And the cell membrane, the double layer, is made up of phospholipids. So one layer is hydrophilic, and one layer is hydrophobic. Hydrophilic literally means loves water, and hydrophobic means hates water. The hydrophobic part is made of fat, which you know resists water. The nucleus of the cell, which is located in the soma, is kind of like the executive office of the cell. It contains DNA, which is the blueprints for making proteins, chromosomes, which are double-stranded molecules of DNA, and then genes, segments of this DNA. And there's a nice picture up here, and then one down here as well to show that the relationship between chromosomes, DNA, and genes. And then importantly, nucleotides are the building blocks that make up DNA, adenine, thymine, guanine, and cytosine, which are typically referred to or abbreviated as just ATGC. And just some brief information about proteins for anyone that might have forgotten from the last time they took biology. They are folded up polypeptide chains that serve a particular function in the body based on their shape, their structure, and they can change shape and combine with other proteins. They can be transported from one neuron to another through the axon. Proteins embedded in the cell membrane, as I mentioned just before when we were talking about the cell membrane, form passageways or gates, and these are called, they're called channels, gates, and pumps, and they regulate the flow of substances in and out. There's also a receptor site, as you can see up here, which is like a little groove in the protein, and they're sort of analogous to a keyhole. So if the right molecule comes in, then it might change the shape of the protein. Okay, so moving on to section 3, genetics. The important that you know the difference between a genotype and a phenotype. The genotype is the genetic makeup of an individual, so the actual genes that you're born with, and a phenotype is the observable physical and behavioral characteristics of an individual, and these results from the interplay between the genes and the environment. We're going to talk in this chapter mostly about Mendelian genetics. It's the basic laws of genetics, but it's not the whole story. And you might remember that Gregor Mendel is the father of the science of genetics. He was kind of the first person that described genetics in the way that we basically understand it now. He was this, I think, Czech monk who was really into his pea plants. Okay, so some basics of genetics. The nucleus in each human somatic cell contains 23 pairs of chromosomes, so 46 chromosomes in all. The chromosome pairs are numbered 1 through 23. Autosomes are is what we call the chromosomes in pairs 1 through 22, and then the 23rd pair are sex chromosomes, so that's the X and the Y. And typically males have XX chromosomes and females have XY, but there are, of course, deviations to this. One member of each pair, so one chromosome from each pair comes from the mother, and one chromosome of each pair comes from the father. An allele is a version of a gene, and a gene pair contains 2 different alleles. If both alleles in a chromosome pair are the same version of that gene, they're homozygous, and if those alleles in the pair are different, they're called heterozygous. Alleles are expressed in different patterns. One pattern is complete dominance, where only dominant gene is expressed and the recessive gene is completely suppressed. Incomplete dominance is when each allele's trait is only expressed partially, and codominance is where each allele's trait is expressed fully. Okay, genetic mutations. These are errors in copies of a gene that create altered alleles. So it can be a change in a single nucleotide, for example. So one of these A, T, G, or C nucleotides, and that creates a new single nucleotide polymorphism. Genetic mutations can be a blessing or they can be bad. So there's the hemoglobin mutation that causes sickle cell anemia is bad, but then a different mutation or a different version of this gene can be good. It actually offers resistance to malaria. Okay, so one example of a genetic disorder that affects the brain, we'll talk about a recessive one and a dominant genetic disorder. So the recessive, an example of a recessive genetic disorder is Tay-Sachs disease, and this is, in this case, a dysfunctional protein causes buildup of fats in the brain, and the individual dies, I think, within 5 years of life at maximum, typically. A baby who is homozygous for recessive mutation of this gene will have severe motor and cognitive impairments. Because this gene is recessive, both parents have to be carriers, and if both parents are carriers, if you look at this figure on the right, there's a 25% chance that the offspring will inherit this disease. Okay, so if you see over here, carriers for the disease, interestingly, have protection against tuberculosis. So if you have one dominant and one recessive allele, which means you would not have the disease, but you would be a carrier, there's kind of an evolutionary advantage to being a carrier, which might explain why this disease is still present in our gene pool. An example of a genetic disorder that is dominant is Huntington's. So most of you are probably familiar with this. It's caused by an abnormal version of the Huntington protein, which kills brain cells, especially in the basal ganglia and the cortex. If you remember, the basal ganglia has a range of functions. The Huntington's disease, one thing you can observe that's kind of marked is this chorea, which means like dancing, and it's an involuntary movement of the limbs, and you can look at this YouTube video of an individual who has Huntington's chorea. The allele for this gene, for this disease is dominant. And if you look over on the right here, you'll see that that means that offspring who have it, offspring with one parent who has it, there's a 50% chance of getting the disease. If there are two parents who carry the dominant gene, there is a 75% chance of inheriting the disease. But notably, only one copy is required to inherit Huntington's disease. Okay, moving on to chromosomal abnormalities. There are two main ways that chromosomes can be abnormal. One is a deviation in the copy number variance. So too many or not enough copies of a disease can cause too many or not enough copies of a gene can cause issues. Several psychological disorders such as autism, schizophrenia, and some learning disabilities are associated with copy number variance, changes in copy number of part of a chromosome. An entire chromosome can also either be transmitted as a pair, as a double, or not enough. So Down syndrome is an example of a situation where there's one extra chromosome. So the 21st chromosome is copied, I think, typically on the mother's side, and it causes a range of issues. Probably everyone is familiar with some of the cognitive problems, motor problems, and cardiovascular issues associated with Down syndrome. Quick note, don't worry about the section on genetic engineering. It is definitely interesting, but it's not directly applicable to things we'll talk about later, so you can read it if you're interested, but don't worry about studying that part. Okay, so all this talk of genes, it's important to note that genes alone do not determine a person's phenotype. Otherwise, two people with the same genes, such as identical twins, would look and act exactly the same. So some examples from my personal life: I have an identical twin sister. She's here on the right, and she and I have very different phenotypes, both behaviorally and physically. We've always looked very different, and we also have quite different preferences. She's an artisan cheesemaker, and she lives on a farm and is great at cooking, and I am obviously in academia, and I am terrible at cooking. So we're quite different. And then I couldn't resist. Here's another example. These are my two baby nieces. They are identical twins as well, and even at 7 months old, they're already quite different. So Lucy over here likes carrots, and she likes reading books, while Eva does not like carrots, and she likes playing with her big brother. And they also look rather different. They look different enough that that you can tell the difference between the two. So even after 7 months, the environment has interacted with their genetics enough to differentiate the two. So phenotypic plasticity is the ability of the genotype to produce more than one phenotype. Epigenetics is the fact that an organism's environment can change how a gene is expressed. This doesn't mean that the actual genes are changed. There is kind of a next level of chemicals that can turn a gene on and off. And this is another way in which the environment affects our phenotypes. There's been a few cases in which scientists have noted that we can kind of inherit some epigenetics. So organisms might inherit some of these we call them epigenetic tags from their parents. which affect how the genes are expressed. Again, the genes aren't actually changed, but they're kind of tagged by certain chemicals that turn them on and off, and so some of this can be passed through from parent to offspring. And this has been likened to inheriting the experience of your parents. One notable example is that in Israel, they did a study where they found that Holocaust survivors may have passed on susceptibility to stress due to epigenetic changes during their traumatic childhood. So these were Holocaust survivors who had been in concentration camps and, of course, had extremely traumatic childhood experiences. This changed their own epigenetics. It changed the way that the Holocaust survivors' genes were expressed. And there's evidence that they passed some of these epigenetic tags on to their offspring, such that some of the children of Holocaust survivors have a higher risk for developing PTSD or a higher susceptibility to stress. Notably, there have been some studies that also indicate that while some children of Holocaust survivors have a decreased resilience to stress, some also have increased resilience. So it's a relatively new field of study, and hopefully some more clarity will come over time.